Have you ever really thought about how life-saving medicines like vaccines or antibody treatments are made perfectly safe to be injected into our bodies? Well, hidden deep inside pharmaceutical manufacturing lines are these incredible invisible guardians, these microscopic gatekeepers that handle one of the most critical safety steps in all of modern medicine. Let's get into it. So here's what we're going to cover. First, we'll tackle what's called the sterile drug dilemma. Then, we'll meet the microscopic gatekeeper that solves it. We'll look at why it's such a vital safety net for pharma, run through the gauntlet of tests they have to pass, and finally, check out the massive multi-billion-dollar market behind it all.
All right, let's jump right in with the core problem, the sterile drug dilemma. It all comes down to this. What do you do when you can't use heat to sterilize something?
This is really the one question that drives this entire technology. How do you sterilize something that you can't heat up? We all know heat is great at killing germs, right? Think of an autoclave in a hospital. But what happens when the very medicine you're trying to make safe is also incredibly delicate?
And that's the real challenge. A lot of today's most advanced medicines, we're talking about biologics like vaccines and complex antibody therapies, they're extremely sensitive to heat. If you were to try and sterilize them with high temperatures, these intricate molecules would literally just fall apart. They'd be destroyed. So you're stuck, because sending out a non-sterile injectable product isn't just a minor quality issue, it's a huge, life-threatening risk to patients.
So, what's the solution? Well, that brings us to the hero of our story, the microscopic gatekeeper. It's really an elegant solution to a very high-stakes problem.
So instead of trying to kill the microbes with heat, sterile filtration does something totally different. It physically removes them. The best way to think about it is like an unbelievably sophisticated sieve. You pass the liquid medicine through a special membrane that's full of microscopic pores, and the principle is just brilliant in its simplicity. It separates, it doesn't kill. The bacteria and other microbes are just too big to squeeze through the holes.
So just how tiny are we talking? 0.22 micrometers. That is the magic number. It's a size that was very specifically chosen because it's smaller than the smallest known bacteria, which basically guarantees they get blocked and can't get into the final product.
And where does this all happen? In a big manufacturing plant? Well, as you can see here, it's right at the very end of the line. After all the complicated biology and purification steps are done, these filters act as the final gatekeepers. They are the last check to make absolutely sure the drug is sterile right before it gets filled into its vial.
It's funny. That 0.22 micrometer standard wasn't always the rule. For a long time, the industry actually used 0.45 micrometer filters, and everyone thought that was good enough. But then, back in the 1960s, a researcher discovered this one tiny bacterium, Brevirdemonas diminuta, that could actually slip through those larger pores. That discovery, as you can imagine, completely changed the game and led to the much stricter standard we rely on today.
Now, this technology is obviously essential, but like anything, it's not perfect. It comes with its own set of trade-offs. So let's weigh the pros and cons that make this pharma's indispensable safety net.
Okay, the advantages are just massive. It's really the only way to sterilize a lot of these modern drugs, and it adds this crucial final layer of safety. But on the flip side, it does make things more complex. You always have to account for the potential that a filter might fail, which means you need to do a ton of rigorous testing. And from an engineering standpoint, it can create issues like a higher pressure drop in your system.
So how do you manage that risk of a filter failing? Well, the industry uses a strategy called redundant filtration. It's basically a backup plan built right into the system. They put two sterilizing filters in a line, one right after the other. The idea is if the first filter has some kind of tiny flaw, the second one is there to catch anything that might have gotten through. This is so important that it's actually recommended by both the FDA in the US and the regulators in Europe.
And that leads us perfectly into our next topic, because a filter is only useful if you can prove it's working. So, welcome to the Gauntlet of Integrity. This is all about proving that the filter is physically intact and doing its job perfectly, every single time.
And the stakes here are just incredibly high. We're talking about the physical wholeness of the filter membrane. A single microscopic flaw, a tiny tear or an oversized pore could let bacteria through and compromise an entire batch of medicine, a batch that could be worth millions of dollars. That's why it's an absolute rule. You have to test the filter's integrity both before you use it and after you use it, every single time.
So how do you even find a flaw that's too small to see without destroying the filter in the process? Well, with some really clever non-destructive tests.
The bubble point test, for example, wets the filter and then measures how much pressure it takes to force gas through the pores. If bubbles show up at too low a pressure, you know you have a flaw. Other tests, like the pressure hold or diffusive flow, are basically super sophisticated leak checks. They're all asking the same critical question: is this barrier intact?
And this all gets put into practice with a super strict step-by-step procedure. Before a batch run, they test the first filter, F1. If it passes, great, they move on and test the second filter, F2. If either one fails, they have to stop and figure out what's wrong. And if a filter keeps failing the test, the whole process is halted, the filters are replaced, and a full-blown investigation is launched. It's a perfect real-world example of good manufacturing practice.
Okay, let's zoom out for a minute. We've been focused on the microscopic level, but let's look at the big picture. In our last section, we're going to explore the massive global industry that's been built around this tiny technology. This is the multi-billion-dollar market.
The scale here is just staggering. If we look back at 2019 data, the total market for all these kinds of biopharma filters was over $8.6 billion. But look at this. Sterile filtration is the biggest piece of that pie by far, coming in at $4.7 billion. That really shows you just how fundamental this one step is to the entire biopharmaceutical industry.
And this isn't some sleepy, static market either. It's growing, and it's growing fast, at over 10% per year. Why? Well, that growth is being directly fueled by the rising demand for all those advanced biologics we've been talking about. It's pretty simple. The more complex drugs we develop, the more sterile filtration we need.
This whole space is really dominated by a few huge global players. You might recognize names like Pal, Sartorius, or Merck Millipore. They don't just sell one kind of filter, they offer a whole spectrum of products for every stage of drug production. It's a very specialized, very high-tech field.
So, if there's one thing to take away from all this, it's that in our world of mind-blowingly complex biotechnology, where scientists are literally engineering cells and molecules, one of our final, most critical lines of defense comes down to a simple, elegant physical barrier. A sieve, basically. Which kind of leaves you with a final thought, doesn't it? What other seemingly low-tech ideas are out there, quietly and reliably keeping our modern, high-tech world safe?